Okay, so you might remember pedigree charts from ninth grade, but basically, what are pedigree charts? They're almost like family trees, but they're tracking the inheritance of a particular trait as well. So they show the relationships with individuals, but they also show the inheritance patterns of traits. And so, how do we identify pedigree charts, or how do we identify individuals? Well, here's a little key over here. Circles are representing females and squares are representing males. If the circle is filled or the shape is filled in, those individuals are affected. The non-shaded shapes are the individuals who are not affected. And so if they have a line connecting them like these two do, that's representing a mating pair. So this would be the mom and dad. All right. And then if they have lines connecting from the top, like individuals C and D, that represents that these two are siblings and they are children of A and B. So how do we figure out the genotypes and the phenotypes of these individuals? So the genotype of individuals A and B, and remember, we're looking at cystic fibrosis still. So cystic fibrosis is recessively inherited. So individuals A and B do not have cystic fibrosis. However, they do have a son who has cystic fibrosis, which means that the son's genotype has to be little t, little t. And remember that one allele comes from mom and one allele comes from dad, which means that mom and dad must both be carriers. But what about individual T? So individual D. Individual D must also be heterozygous. And so how do we know that? Individual D married someone who has cystic fibrosis. They also, if we look at their children, have a child with cystic fibrosis. So this individual H, has to be little t, little t. And remember that one allele comes from mom and one allele comes from dad. So in that case, mom, D, individual D, must have at least one little t. And so they must be heterozygous. All right, and then sometimes we'll ask questions about individual D and his mate. So that's these two right here. So individual D is actually a female. So individual D and her mate are planning to have another child. Calculate the chances of the child having CF. So remember, we already determined that individual D is going to be heterozygous, big T, little t, and we know that her mate is little t, little t. So when we set that up, we would get a Punnett square that looks like this. And so their children have a 50% chance of having cystic fibrosis and a 50% chance of being carriers of cystic fibrosis. These are some other examples of autosomal recessive diseases that you may have heard about. So albinism, that has to do with the lack of pigmentation produced in our skin. All right, phenylketonuria, also known as PKU. Tay-Sachs disease is also another autosomal recessive disease, as well as thalassemia. And so just take a look over here. It says genetic diseases are very rare. So even the most frequent occurring autosomal recessive disease only affects 1 in 2,000 people, but most others are more along the lines of 1 in 10,000 or 1 in 20,000. Okay, so another example of a recessive, or sorry, actually this is codominant, is sickle cell anemia. So sickle cell anemia is caused by a base substitution mutation. And so what that means is that one base in the DNA of this particular gene was substituted or accidentally swapped for another. And so if we look, this has to do with the hemoglobin gene. And we know that hemoglobin is a protein that we find in our red blood cells that helps us to carry oxygen in our red blood cells. And so if we take a look at the DNA, this is supposed to be the sequence of the DNA, but the T right here was accidentally swapped or substituted for an A. Last unit, we talked about how the change in the DNA is going to result in a change in the mRNA, which could result in a change in the amino acid sequence. And that's what we're seeing here. If you notice, the mRNA is supposed to be GAG, which codes for glutamic acid, but because of the mutation, the mRNA that is transcribed is GUG. And so this GUG codon codes for valine instead. And so that change to valine is going to have an impact on the shape of the hemoglobin protein, and therefore it's not going to function properly. So when sickle cell anemia is inherited, this abnormally shaped hemoglobin is more fibrous than it should be. So hemoglobin should be more globular, but it's more fibrous, which you can see here as well. So what happens is, because of the shape of the hemoglobin protein, well, first of all, it can't carry oxygen as efficiently, the red blood cells, and the red blood cells also become this sickle or curved shape. That could also cause blockages in the capillaries. All right, but mostly people feel symptoms such as weakness, fatigue, shortness of breath, okay? However, there is some benefits to sickle cell anemia, or at least having the trait for sickle cell anemia. Because remember, sickle cell anemia, we're going to look at the inheritance patterns, but you can be a carrier of sickle cell anemia and still not have sickle cell anemia. So malaria, you may have heard of malaria. It's a parasitic disease that is carried by a specific type of mosquito. And what happens is that this parasite invades normal red blood cells and causes them to lyse, or basically to break. So the symptoms include fever, shivering, vomiting, and anemia. Death is common, often due to kidney failure. But what scientists have noticed is that malaria cannot infect sickle cells. So people with sickle cell trait are resistant to the disease. And what they've noticed is that sickle cell is more prevalent in areas where malaria is found or more common. So let's take a look at how sickle cell is inherited. So sickle cell is an example of genetic inheritance due to codominance. And so remember when we talked about codominance in our last lesson, all right, a lot of times we use the superscripts to represent codominant alleles because the codominant individuals have a mixed phenotype. Okay, so we're going to use these A and S superscripts, A representing normal hemoglobin, S representing sickle hemoglobin. Okay, and so if we look at these genotypes, there's 3 options, just like any other particular gene or trait. So if you have 2 normal alleles for hemoglobin, all right, and you are homozygous for normal hemoglobin, the phenotype is normal. All right, and so these people have normal red blood cells and normal hemoglobin and are not protected against malaria. All right, but if you are heterozygous, remember when we're talking about diseases, these individuals we refer to as carriers. So this individual has 1 normal hemoglobin allele and 1 sickle hemoglobin allele. So these individuals do produce some sickle cells. However, they're not really affected by the disease because they are producing normal cells as well. And so that's why it's considered codominance. But these individuals are also protected against malaria, which is interesting. So if you are homozygous for sickle hemoglobin, all right, these are the individuals that are affected and have the sickle cell disease. They're also resistant to malaria. All right, but remember, the heterozygous individuals are carriers and do not have sickle cell anemia. So we're going to use our Punnett squares, and so let's predict the phenotype in this cross. So remember that sickle cell is codominant, so we're crossing 1 carrier with an affected individual. So a carrier would have one normal hemoglobin allele and a sickle hemoglobin allele. So we're going to put them over here. All right, and then the affected individuals are the individuals with sickle cell anemia. So they have to have two sickle alleles. And so when we do our cross, all right, we would end up with 50% of our offspring, 50% chance that our offspring will be carriers and not have sickle cell anemia. But 50% of the offspring, or there's a 50% chance that the offspring will have sickle cell anemia. So another type of disease that we need to talk about is Huntington's disease. And so Huntington's disease is a little bit different because it is a dominant allele. It is still an autosomal disease, all right, because the gene for Huntington's is located on chromosome 4, all right, but this is dominantly inherited. And so what that means is if you are a carrier of Huntington's, meaning they are heterozygous, then they still have Huntington's, because remember, it's dominant, and the dominant is going to mask the recessive. So what is Huntington's disease? So basically it causes severe nerve damage. And oftentimes these symptoms don't show up until someone is about 40 years old. So once they start their symptoms, they may have difficulty walking, speaking, holding objects. They may experience shaking in their limbs. And unfortunately, within a few years after symptoms start to show, the person could lose complete control of muscles and die of an early death. So this is a fatal disease, but we won't see it start to onset until someone is in their 40s or midlife. And again, this is caused by a mutation in the gene on chromosome 4. All right, so if you look, this is the normal gene and it's producing a normal protein, all right, but this would be the abnormal protein. And so when abnormal proteins are produced, they're not doing their functions properly. And this protein being produced because of the gene is going to cause degeneration in the brain and the nerves. Okay, so let's take a look at the inheritance of Huntington's. So remember that if you look over here, here's our key. So remember that Huntington's is now a dominant disorder. And so the recessive individuals are going to be the normal gene. So what's the probability of an unaffected mother? So here's our unaffected mother, the homozygous recessive individual, crossed with a heterozygous father for Huntington's. So remember that this heterozygous father has Huntington's disease. So we set up our Punnett square, and you can even pause this video and see if you can do this on your own. And so what do we get? So there's a 50% chance of having a child with Huntington's disease. Those would be these two individuals. All right, and there is a 50% chance of having a child that is not affected by Huntington's disease. So I think we spoke about when we were talking about in vitro fertilization and how they could potentially screen the embryos before implanting them into the mother's uterus. This would be an example of why they might do that. If they know that they have a genetically inherited disease and they want to have children, they can screen the embryos for embryos that do not have the disease and then only insert those embryos into the mother's uterus.